Welcome to our journey into the fascinating world of animal diversity. Today, we're looking at the phylum Porifera, specifically the canal system. Imagine being glued to a rock at the bottom of the ocean, unable to move, hunt, or even chew. How would you survive? Sponges have solved this problem brilliantly by turning their entire bodies into highly efficient, living water pumps. Let us dive in and see how this incredible biological machinery works. Now that we understand the sponge's basic problem of being stationary, let us look at its solution. Because sponges cannot walk or swim to find food, and they completely lack a mouth or stomach, they have to bring the ocean to them. They do this by acting like a giant biological vacuum cleaner. Water is constantly pulled in through the sides of their bodies, filtered for tiny food particles, and then blasted out. This continuous flow of water is the absolute most important feature of a sponge's existence. So, how is this biological vacuum cleaner structured? In biological terms, we call this the aquiferous system. It is a complex network of channels. The water first enters through thousands of microscopic pores on the outer surface, which are called ostia. From there, the water flows into a large central cavity inside the sponge, known as the spongiocele. Finally, after the water has been filtered, it exits the sponge through a large chimney-like opening at the very top, called the osculum. We know the path the water takes, but what actually pumps it? The entire system is powered by specialized cells called choanocytes, or collar cells, which line the inside of the sponge. Each of these cells has a tiny whip-like tail called a flagellum. These flagella do not just flap randomly, they beat back and forth in a highly coordinated rhythm. This synchronized beating creates a strong, unidirectional water current that constantly pulls water into the ostia and pushes it out through the osculum. As sponges evolved, their canal systems became more advanced to support larger body sizes. Biologists classify these systems into three main evolutionary grades. The simplest is the asconoid grade, which is basically a straight, hollow tube. Next is the syconoid grade, where the body wall folds inward and outward to create more surface area for filtering. Finally, we have the leuconoid grade, which is the most complex. It features a highly branched network of canals and chambers, maximizing the sponge's ability to extract nutrients from the water. Let us look at these three grades visually. On the left, you see the asconoid system. Notice the simple, straight walls where water flows directly into the large central spongiocele and out the top. In the middle, the syconoid system shows folded walls, creating small pockets that trap more water. On the right, the leuconoid system is vastly more complex. The central cavity is reduced, replaced by a dense maze of tiny chambers lined with choanocytes. This massive increase in surface area is what allows leuconoid sponges to grow to massive sizes in the ocean. Now that we have seen the structure, why is this system so critical? Because sponges are incredibly simple animals. They lack true tissues, organs, a nervous system, and gills. Therefore, the canal system must do everything. As water flows through, it brings in dissolved oxygen for respiration and microscopic plankton for food. Simultaneously, the outgoing water sweeps away carbon dioxide and metabolic wastes. It even handles reproduction by carrying sperm out into the ocean currents and drawing in sperm from other sponges. It is a multi-purpose survival engine. To truly appreciate this biological engine, We must look at the physics behind it. Sponges perfectly exploit the principle of continuity. This principle states that the volume of fluid entering a pipe must equal the volume leaving it. In our formula, A in is the total area of all the tiny pores, and V in is the speed of the water entering them. A out is the area of the large exit hole, and V out is the exit speed. Because the combined area of millions of tiny pores is huge, water enters very slowly, allowing time for feeding. But as it is forced out of the smaller exit hole, the velocity must increase. Before we move on, let us see this with a concrete example. A marine biologist is studying a sponge where the total pore area is 100 square centimeters, and water enters at a slow 0.1 centimeters per second. The exit hole is only 2 square centimeters. We want to find the exit velocity. We start with our continuity equation and plug in our numbers. Multiplying the left side, we find 10 cubic centimeters of water enter every second. To find the exit speed, we divide this flow rate by the exit area of 2. The result is 5 centimeters per second. The water shoots out 50 times faster than it entered, blasting waste far away. That high-speed jet of wastewater is just one part of the story. Sponges are also ecological powerhouses. Because those tiny choanocyte cells never sleep and never take breaks, they are pumping water every single second of the day. This constant filtration cleans the ocean water, which is vital for the health of the entire surrounding marine ecosystem. To truly grasp the scale of their work, we can calculate exactly how much water a single sponge processes over a full day. This brings us to our next mathematical tool, the standard volume formula used in fluid dynamics. The formula is V equals Q times T. Here, V is the total volume of water the sponge filters. Q represents the constant flow rate, which is how much water moves through the sponge every second. Finally, T is the total time the sponge is pumping, which must be measured in seconds to match the flow rate. If we know how fast the sponge pumps and for how long, we can find the total volume. Let us apply this to our previous sponge, which had a flow rate of 10 cubic centimeters per second. We want to know how many liters it filters in one full day. First, we must convert 1 day into seconds by multiplying 24 hours by 60 minutes and 60 seconds, giving us 86,400 seconds. Next, we multiply this time by our flow rate of 10. This gives a massive 864,000 cubic centimeters. To make this easier to visualize, we divide by 1,000 to convert to liters. The final answer is 864 liters per day. A single small sponge filters hundreds of liters daily. We have covered a lot of ground today. Let us review the key takeaways. First, the canal system is the defining feature of sponges, acting as a multi-purpose biological engine since they lack true organs. Second, this system is entirely powered by the coordinated beating of microscopic choanocyte cells. Third, sponges brilliantly apply the physics of fluid dynamics, ensuring water enters slowly for feeding and exits rapidly to blast away waste. Finally, these simple, stationary animals are incredibly efficient pumps that have survived for millions of years while keeping our oceans clean. Great job following along!